The eye is a sensory organ that can function as a camera, in that the aperture of our own eye is the pupil, which controls the amount of light that enters, varying on the environmental light intensities to help form the clearest image. Light is an electromagnetic wave that passes the lens of the eye, and with the help of accommodation by the lens, light reflected from objects can be absorbed by the retina, which is then transduced into electrical signals that fire as action potentials from the eye to the brain to allow us to see. Light enters the eye through the pupil. The lens shape changes depending on whether the object we are looking at is near or far away. Distant objects reflect light rays that require less refraction, therefore the lens does not need to be as wide and thick. Here, distant object light rays enter the eye and the ciliary muscles relax, increasing the tension on the zonual ligaments holding the lens in a thinner, flattened shape. This reduces the refractory power of the eye, and the light rays can be converged onto a focal point on the retina. For near objects, the ciliary body contracts and the zonual fibres have released tension, allowing the lens to be short and wide to increase its refractory power. This again allows the light rays to converge onto the back of the retina. Sometimes problems arise when the accommodation does not work properly. People may experience hyperopia, otherwise known as farsightedness, and these people can see distant objects fine, but close objects are focused onto a point beyond the retina. Other people may have myopia, short-sightedness. Here the light rays converge at a point before the retina, and they are unable to view far objects clearly, but can observe close objects fine. Hyperopia can be corrected using convex lenses, whilst myopia can be corrected using concave glasses lenses. At the centre of the retina is the fovea. The fovea is the thinnest part of the retina and contains the cone photoreceptors, which are responsible for central vision. Cones have 3 different photopigments, and this allows for our coloured vision, whilst rods are found at the periphery and are in a much higher abundance and facilitate scotopic, otherwise known as night vision. Light energy is converted into electrical energy for the brain via phototransduction. Rods and cones contain different photopigments, and they are found in the outer discs of the photoreceptors. Rods contain rhodopsin as their photosensitive pigment, and it consists of retinal and opsin, and when in the dark, they remain associated unbleached in the discs of the photoreceptor. This allows guanylyl cyclase to produce CGMP, which opens cyclic gated nucleotide channels to allow calcium and sodium influx. This depolarizes the membrane potential to -30 millivolts, so the net influx exceeds the potassium efflux, which allows exocytosis of glutamate onto the off bipolar cells. Off bipolar cells are active when in the dark because of the high glutamate, and are active in the light because of reduced glutamate levels. These bipolar cells send this action potential to ganglion cells, modulated by horizontal cells, of which the axons of the ganglion cells bundle together to form the optic nerve, which leaves the eye via the optic disc and crosses over at the optic chiasm, which follows the optic tract and sends this information to the lateral geniculate nucleus of the thalamus, which can then be relayed to the striate cortex of the occipital lobe to form an image. When in the light, the rhodopsin is bleached, leaving the bleached opsin in the disc and the activated retinal to dissociate into the retinal pigment epithelium, where it can then activate transducin to stimulate phosphodiesterase to inactivate guanylyl cyclase, which reduces the production of CGMP, which closes the cyclic gated nucleotide channels, and now the potassium efflux exceeds the net positive influx, which results in hyperpolarization, inhibiting the release of glutamate. This activates on-bipolar cells, which send signals as described before. On-bipolar cells are active in low glutamate conditions, so in the light, and are inactive in the dark, where there are high glutamate levels. Glaucoma is the leading cause of blindness. Risk factors include race, genetic predisposition, and smoking. And there are 2 main types: closed angle and open angle. Glaucoma causes increased intraocular pressure through inadequate aqueous humor outflow and increased aqueous humor production. The aqueous humor is produced by the ciliary body and is distributed into the posterior chamber behind the iris. It is supposed to be able to leave through the trabecular meshwork and into Schlemm's canal or through the suprachoroidal space. The increased intraocular pressure results in compression of the optic nerve, which causes degeneration of the retinal ganglion cells and ultimately leads to vision loss. Treatments of glaucoma aim to reduce the production or increase the aqueous humor outflow. Prostaglandin analogues like latanoprost and travoprost work to increase the uvuloscleral outflow. Beta blockers are only indicated for chronic glaucoma and include betaxolol and timolol. They work to reduce the production of aqueous humor. Alternatively, someone may opt for an alpha-2 agonist. M3 agonists like pilocarpine and carbachol work by constricting the pupil, which physically opens the trabecular meshwork, increasing the outflow. Age-related macular degeneration is the leading cause of blindness in those over the age of 65. It results in central vision loss. This occurs due to the loss of photoreceptors at the macula densa of the fovea as a result of lipofuscin accumulation, which contains lysozymes at the retinal pigment epithelium, which results in altered metabolism of the degraded photoreceptors. This causes the accumulation of drusen deposits under the retinal pigment epithelium within Bruch's membrane, which contributes to the retinal pigment epithelium detachment. This reduces fluid movement from the RPE to the choroid. There are two main types of age-related macular degeneration: wet if vascular and dry if not. Dry can become wet. Treatment for wet age-related macular degeneration involves the use of monoclonal antibodies against vascular endothelial growth factor. An example would be bevacizumab, which inhibits angiogenesis. And for dry age-related macular degeneration, drugs are aimed at reducing the inflammation. Examples may include pegcetacoplan, the C3 protein inhibitor, non-steroidal anti-inflammatory drugs, visual cycle modulators like synthetic vitamin A because it produces toxic vitamin A dimers more slowly than endogenous vitamin A. Additionally, lots of work is being done into stem cell replacement therapy, as this could potentially replace the lost retinal pigment epithelial cells.